Thank you all for coming. It's an absolute pleasure and a privilege to introduce today's speaker, Peter Currie, who's the director at the Australian Regenerative Medicine Institute down at Monash. Peter's world-renowned for his work in muscle stem cells and muscle regeneration with a model organism of the zebrafish. And I think you'll see today from the quality of the science that he will be presenting, if it's anything like what I've seen in the past, which I believe it is, I think you'll be as blown away by it as I am, and it'll be very clear to you why it is that he is frequently published in journals like Nature, Science, and Cell Stem Cell. Peter's background was that he obtained a PhD from Syracuse Uni in New York City, or New York State probably, postdoc at Cancer Research UK, and has had lab head appointments at the Medical Research Council in Edinburgh, but also at the Victor Chang Cardiac Research Institute up in Sydney. It was only a couple of years ago, 2016, that Peter was appointed as the director at ARMI, and has continued his work on, his beautiful work on stem cell work and muscles since then. Peter has received awards from the EMBO Young Investigator Award, is an SPRF, a senior fellow with the NHMRC, Wellcome Trust Research Fellowship, and the President's Medal for the Australian and New Zealand Society for Cell and Developmental Biology. So, Peter, thank you very much for taking the time to come and talk to us. Very much looking forward to it.
Okay, great. So today we're going to talk about fish and how fish make their muscles. So my lab is completely fascinated, or if they aren't when they start, they have to be by the time they finish, with this structure here, the embryonic myotome, the way that it's made. So my lab chiefly works on way too many things. I actually had the great pleasure of going to wish farewell to one of my mentors in Edinburgh, who first gave me my first lab head position at the start of the year, and he got up to introduce me at my seminar, and he said, "Currie, you still work on too many things," which is absolutely true. And actually, these have been core themes within my group. But what holds everything together, like it does in this anatomist drawing, is the fact that they're all tied to one organ, the musculature. So I cut my teeth on specification and morphogenesis. We work in differentiation and disease of the muscle. I'm endlessly fascinated by evolution, so if you want to talk about that, set aside a couple of hours. But today we're going to talk about stem cells, and we're going to talk about those two different sets of stem cells and basically a series of serendipitous findings. But I want to use these to illustrate some general principles in regeneration biology and set the stage of some of what I think are the bigger questions that as regeneration biologists, we're faced to tackle. So here is my favourite artist, John Singer Sargent. This is actually a ceiling motif, but here he's illustrating one form of regeneration biologist. He's Hercules with a Lernian hydra, and I don't know how quite he does it with his club, but when he cuts off the head, another two regrow. So in classical literature, of course, regeneration is either seen as a punishment, as Prometheus and the regenerating liver with his eagle, or a fascinating but somewhat scary property of a unique set of organisms, and here's the Lernian hydra. Of course, hydras we work with these days, not quite so scary as that. So, and this hydra here has what we would consider the Rolls-Royce ability of regeneration, that you could make all your tissues from a single cell. Now, my good friend John Moman, who's a plant biologist, always reminds me how pathetic animals are at regeneration, because he says that every cell in a plant is a stem cell. So top that if you can. But again, a hydra has that property, and it also is able to make complex morphological structures no matter how you slice and dice it. So this is called epimorphic regeneration, the ability to create any tissue and organ and any structure from an individual cell that dedifferentiates into a pluripotent state, not that different to what we experience in our early life, to produce tissues and organs. There are other regenerative capacities that are exhibited by other organisms, and here we are illustrating one that I'm particularly fond of. So this is the zebrafish, and it is not unique amongst vertebrates. There are many vertebrates that have these capacities, but it can regenerate many of its tissues and organs. In this movie, we're showing one of perhaps the most famous characteristics of de-differentiation regeneration, which is allied or akin to the epimorphic regeneration that you see in a hydra. But it's a tissue-specific de-differentiation event. So if you or I were to have a myocardial infarction, of course, this would result in death of the myocardium at the site where the ischemic event occurred, and you would never recover that, and the heart would remodel it. But in a zebrafish, if you go in and if you resect up to a third of its ventricle, don't do this at home, by the way, it forms a scar just like we do initially, and then the epicardium, the cells around it, come to surround the scar. There are signals that are secreted at the interface and also from the epicardium. But over the course of around a month and a half's time, the cells of the epicardium regenerate, and actually they de-differentiate. That's my good friend and colleague Kazu Koyuchi up at Victor Chang had an amazing Nature paper about 6 years ago to show that the de-differentiation occurs within the cardiomyocytes themselves, and they regrow and repopulate and remove the scar and remodel. And signals from the epicardium are secreted that direct aspects of this, and the scar is removed. So from this we know that some animals can regenerate and some animals cannot. And there's a panoply of the way that cells regenerate. So mammals, us, still have some regenerative capacity. So thank God our liver regenerates. I think half of the weehive would not be here if that was not true. At least that's what I've heard. Blood, of course, this is what many in the room have cut their teeth on, the fact that blood is a regenerating system, and our only, well, not only, but our most effective stem cells treatment. And we know skin, and I'm going to talk to you today about muscle. But you see here, across the phylogeny, a great variety of different regenerative characteristics. And so we have lost many of these particular things. I've just moved this to here. I think this is one of the best regenerating systems. So Ken puts it here, I put it over here. But you'll see we have lost some of the ability to regenerate that other systems have got. Of course, one of the big things is, why is that? Why are we less able to regenerate our organ systems? And what can we learn from those animal systems that have greater regenerative capacity? So this fascinates a large proportion of regeneration biologists. Some think it's allied to the fact that regeneration is a bit like cancer, and of course if you regenerate you might be more prone to cancer. I don't think this is true. Most of these organisms down at this end of the phylogenetic scale have no greater risk of cancer than others. Some, like sharks for instance, are phenomenologically believed not to get cancer, which I also think is bunkum. But nevertheless, I don't think that's true. I prefer the hypothesis that we are just losers and we have lost this capacity. So within every different lineage of the vertebrate clade, there are different capacities to regenerate different tissues. And more fascinating, there are different ways to regenerate those tissues. So I just showed you the heart, which is a striated muscle that contracts, and it regenerates beautifully in zebrafish from the de-differentiation model. And what I'm going to talk about today is muscle here, and here is my organismal system, and how these two systems do it. So would it be the same as cardiac muscle, where you undergo de-differentiation, go back into the cell cycle and proliferate? What an amazing party trick if you could pull that off on all your tissues, wouldn't it be? Or is it more like this system here, which is our own system, which has a resident stem cell system? So these are the two things, these two model systems that we reference and study. So today I'm going to tell you two vignettes of two PhD student theses that have happened over the last 5 years. One of them will be about regeneration. The other will be about how stem cells program growth. And I think growth is one of the last frontiers, and I'll explain why I think that's a really big frontier for developmental and stem cell biology. So David Gurovich, who studies regeneration and other systems, actually the heart, was a PhD student in my group for 6 years ago, and this is 4 years of David's work that I'm presenting. So as I mentioned to you, most of our regenerative capacity, well, all of it's known in mammals, except for some suggestion in the liver, occurs through the presence of resident stem cell populations within different tissues. And skeletal muscle is one example of such a stem cell system, where there are tissue-resident stem cells that respond to regeneration. So when you have a soft tissue injury or corky, or you at the gym and you hyperextend, you activate your stem cell populations. And also in disease such as Duchenne muscular dystrophy and other dystrophic conditions, they're activated to try and replace muscle tissue that's lost. So this is my cautionary stem cell slide. So these particular cells were discovered in 1951 in the muscle of the anurin xenopus. So they've been known a little bit later than hematopoietic stem cells. They're actually simpler than hematopoietic stem cells. They're a single tissue-resident stem cell that resides under the basal lamina of a muscle fibre that have unipotential. They only make muscle. So you've got a resident stem cell in a defined niche, when activated after injury, only produces muscle. Yet we cannot produce, despite the 65 years of study in this relatively simple system, we have not been able to harness them for any therapeutic gain. So you cannot imagine a system simpler than this, conceptually or ideologically, and one assumes, therefore, molecularly. And there are a number of amazing questions as to why this hasn't been. If you take these out of the animal and do transplants outside of the context of their niche, they simply don't proliferate. So my particular predilection is we don't know enough about the niche environment. And why is that? Well, satellite cells are these cells here, and they're the main player. In fact, after being discovered in Xenopus, the satellite cell was more or less hijacked by murine biologists, and actually it was hijacked by the myogenesis crew. So there's a big central dogma around the beta-HLH proteins that were discovered in Mihail Weintraub's group, and the disciples of him have pushed the myogenesis field for nearly 20 years, and they have a very myogenic-centric field of focus. And so they've studied this cell in almost isolation, either by cell sorter populations or single fibre. But of course, that doesn't really recapitulate what a regenerating stem cell needs to see in its local environment. So we wondered whether zebrafish could ever, or provide any, shed any light on any of these questions. But more than that, more basic question was, how does muscle regenerate in a system where you can both de-differentiate cells and possibly have a stem cell system? We didn't even know what system would be used in zebrafish to regenerate. And also, as I'll show you in the second part of the talk, fish continually grow their muscle, so there was an open question about whether you would actually need a regenerative capacity. So we wanted to see if we could image the stem cell in action within the living tissue in a regenerating context. That was the task that I set David. We already knew that they had a regenerative capacity, so a very simple experiment. You take adult fish, you sieve their muscles, or you plate it out, and you see that they have proliferative capacity, and they make beautiful myofibers. There's something in the muscle that can make a myofiber when you take it out that's proliferative. If you simply take the side of a fish and if you take a side of a fish, poke it, you'll see there are highly proliferative cells that come and occupy the wound site and restore muscle integrity, so they're there. So then we wondered how we would tackle this question. So we asked, in a late larval model of injury where we can image, is there de novo myogenesis? So we set up two injury paradigms. I'm showing you one, which is a needle stick injury, which is just what it says. You go in and you stick a needle, a very fine gauge needle, at a particular point every single time. And you use transgenes that mark differentiated muscle here in red, and the stem and progenitor population. This is one of these myogenic regulator factors, MIF5, which marks both the stem and progenitor population. And you can see when you have an injury, you get de novo myogenesis. In fact, this really fairly crude time-lapse was the first ever time-lapse movie of regeneration in vivo. And even just drilling down slightly with higher resolution, we could see there were cells that underwent remarkable. So here's a tissue-resident stem cell. So this is suggesting that they are tissue-resident stem cells, although we have to prove that, of course. And they're activated. Some are not activated and some are. They undergo these incredible elongation behaviors that's never been seen before. They inch into the wound site where they initiate regeneration. It's all characterized by the same genes. You could just put These are all the markers that are used in mammals. So PAC7 is a marker of satellite cells, the stem population. MIF5 is a marker of the stem and activated population. MyoD committed to myogenesis, and myogenin is a myocyte marker. And they all come on in a temporal time frame in these needle stick injuries that support de novo myogenesis. And perhaps most intriguingly, if you look at these regenerating contexts, in mammals, what happens is that you go through a unique phase of differentiation as a muscle cell when you're activated during regeneration. You recapitulate your embryonic myosin expression. So we have different muscles, fast and slow, depending on which muscles are used and what type of athlete you are, fast twitch and slow twitch. And there's embryonic and adult forms of those. But in regeneration, the embryonic form of slow twitch muscles are uniquely always turned on first. And this is recapitulated in the zebrafish, saying that they have a unique de novo myogenic program for regeneration that's different, no matter whether they're in the fast layer in blue or this is the superficial slow layer in green. Right, so then we said, can we image? And again, using very just simple tools, the same MIF5 line and this in green and the differentiated muscle in red. We just made here using the second type of injury, the laser ablation injury, where we just nicked 4 or 5 fibers, and that allowed us to image the entirety of the generative process from the activation of the stem cell in green to the formation of the muscle fiber. Now, there are a number of fairly unique things that we saw that hadn't been seen before, including this enormous filopodial extension and capture by the resonant fibers of the differentiating stem cell, or this point, a transit amplifying cell, against the wound edge. But basically, using this movie, we were able to chart a bunch of unique cellular properties that happened during regeneration. And we charted 4 phases during this regeneration. So perhaps, as I said, most uniquely is the fact that the resident fibers tell the stem cell where they need to go, or the differentiating cell, which of course makes a lot of sense. If you have a wound, the cells around it must tell the regenerating cells, the proliferating cells, the extent of the wound and where they need to fill the wound from. But of course, everyone ever thinks that the stem cell is the intelligent, uni-thinking thing within a stem cell system, which is complete bunkum, of course. It works within a system which includes the uninjured cells themselves. And so this became quite an interesting focus. So the markers we had here are fairly generic markers of myogenesis and differentiation. We wanted to find a marker of the stem cell, and we found one, which is this proto-oncogene c-Met, which is not expressed on the outside of the fish here in section, which is, I'm going to tell you, is the growth zone, which was important for us. And it seems to uniquely mark the stem cell. And when we make double transgenic lines with PAC7, which marks both the growth stem cells as well as the tissue-resident regeneration-specific stem cells, it's a small subpopulation of them. So we use this marker to ask one of the big questions, which is how do cells actually operate, stem cells operate within the wound environment to undergo self-renewal. So self-renewal is this almost magical property of tissue-resident stem cells, capacity to make copies of itself while also providing progenitors for regrowing the tissue that's damaged. Many models have grown a lot out of hematopoietic stem cell biology, but more recently out of skin cell biology. Some favour asymmetric divisions of clonally related and rare stem cells. Some favour a dynamic property between the progenitors and stems themselves that generate stem cells. There's a real emerging panoply of models. And how the stem cell populations in skeletal muscle underwent cell division was an open question. So the on-fibre explant supported a model of asymmetry, but yet there are other evidence in vivo that did not support it. So we wondered whether our model would be able to once for all capture whether an asymmetric division happened. So using the 2 markers I just flashed across the screen, c-Met, which marks a stem cell uniquely, and PAC7, which marks both the stem cell and the transit amplifying compartment, you'll be able to see through these panels that the red cell, the c-Met positive cell, regenerates itself every single time and generates progeny for growth. So we actually caught these asymmetric divisions in real time happening within the tissue. So, as I said, when you think about rare immortal stem cells generating clonal progeny for growth, the big thing that isn't tested in this model, whether it really is truly clonal. And the way that we do this is to create a zebra bow array, which allows us to assay clonality of regeneration. And the way that we do this is to use the zebra bow cassette and drive it from a Semitic specific driver line, which is a Mesogen and Currie driver line, which I've just shown here with the photoconvertible Keita line to show you where it's expressed. So we cloned this Mesogen and drove Currie from it, and this Currie is in every single cell of the somite, which will give rise to every cell of the myotome and give rise to the stem cells themselves. So with the appropriate Currie driver and timing of the Currie, and we can, when combined with the array, the zebra bow array, which is 30 or 40 arrays of these UB-driven cassettes, we can indelibly label every single Semitic derivative in a unique color. And here you'll see a beautiful example of that, where the zebra bow, which we've now called muscle bow, is labeled every single muscle fiber in a unique and different color. So doing that, you can ask, what is the clonality of the regenerative response? So these are the two of the same animals, pre and post injury and regeneration. So here we are at 4 days post-fertilization with no injury. And you can see by going in and ringing every single fiber and ask what color it is, by giving it a value of U on the U and saturans, and then plotting it at a color wheel, you see it's relatively random as to what color these fibers make, which validates the zebra deletion array. However, if you go in there with laser ablation and say, what color is the regeneration, what are color of the fibers that regenerate after injury, you'll see that they're highly clonal in nature and they cluster at a specific site on the color wheel, saying that yes, indeed, rare immortal stem cells give rise to the progeny here that specifically drive regeneration, confirming a long-standing hypothesis from in vitro biology. So these are fairly simple technical things that we've done that have answered long-standing questions in the field, and the ability to access the in vivo biology of stem cell systems is a really strong function of zebrafish biology, but also in a number of systems such as the skin, also in the vasculature and other systems, intervital imaging is emerging in murine systems to counterpoint these studies. And so there are different systems that use different mechanisms for making or replacing cells. So it'll be important to watch this space and see how much consensus emerges about which system uses what and what principles go to what systems. So what I've described here are a series of activities and properties of regeneration of muscle. And we're basically taking all of these, the asymmetric division, the way the cells interact with each other, and amazingly this interaction with the uninjured fibers to affect repair. But how is all this initiated and regulated in the first place? And that is where we're heading. And the really intriguing thing about this particular asymmetric division is at the time when we published our paper, another paper came out indicating that dystrophin itself, so dystrophin is the protein that's mutated in Duchenne's muscular dystrophy. The dogma about the way that disease works was the fact that dystrophin is required for stable muscle attachments across their membranes. The lack of dystrophin causes a disconnect between the dystrophin-associated glycan protein complex and the extracellular matrix. This leads to rupture of the sarcolemma and necrosis of cells. However, Mike Rudnicki's group beautifully demonstrated that dystrophin is actually asymmetrically localized within the stem cell itself. And the dogma about the why you couldn't repair dystrophin after injury was that this constant cycle of degeneration and regeneration exhausted the stem cell niche and it couldn't replace it. But what Michael actually beautifully showed was that there was a problem now with asymmetric division, which we've now shown exists for muscle stem cell biology, on fibers, and what you got was the fact that you didn't undergo self-renewal properly. What you got was a stem cell hyperplasia effect, so that you had more stem cells and very few progeny because this asymmetric division didn't occur in response to extracellular signals yet to be described. So what this work suggests is that Duchenne muscular dystrophy is in part a stem cell disease. And so if you could repair the ability of these cells to correctly divide undergo their asymmetric divisions appropriately, you might be able to restore better regenerative capacity. So of course, the two things in hand produced a fairly powerful and compelling argument about the need to study this process in more detail in vivo. And how do you do that? Well, we've been beginning to try and work out the triggers and the features of the cellular behaviours that coordinate the activation of the stem cell compartment. This is actually an image that was generated by my graduate student downstairs on the WeHi Airy scanner, which I am going to unashamedly put a plug in. It's a beautiful instrument. So if you have cellular biology that you want to study, that is a fabulous instrument. Anyone knows what these cells are? Immune system cells. These are macrophages. So macrophages are cells that come in. They're supposed to clean the wound and go away, whereas these cells are actually, and we've got a lot of data now, we're talking about the single-cell data, beautiful single-cell data on these particular cells. These are the orchestrators of the regenerative response. They uniquely form transient niches here. In pink are the stem and progenitor cells I've mentioned. There appears to be vesicle exchange between the stem cell and the macrophage, and you require them for the activation of the stem cell response. So the nexus between the immune system and regeneration is one of the hottest topics in regeneration biology. Manipulating the immune system in regenerating organisms or ablating it stops regeneration, and it seems to be for a lot more than just the boring reason that you don't clear cellular debris. So we're drilling down on the mechanisms and the molecular pathways that macrophages use to activate the stem cell niche directly. And if anyone's interested in that, there are projects going. Okay, so that's where the field's heading, I think, in vivo regeneration biology. Lots of imaging, lots of trying to understand in toto biology of regenerating systems and understanding the molecular pathways that coordinate activation in a cellular resolution context. Not much difference there, I suppose, to what most people would do. However, as I said, I'm really fascinated by this next subject, how organs grow. And this was the PhD of Fong, who's now in the Ubrecht. And Fong was instrumental in some of the major work that studied the way that the Semitic tissue regulated growth of different systems. But this will be about the musculature. And Fong is actually studying the heart too. I don't know why everyone leaves my lab works on the heart. Anyway, so here we are. So here is what we need to do, I think, as regeneration biologists. Now here are these beautiful little organoids, which you've probably heard a bit about already, if I believe Ed's come to give a talk and other people. We now have the ability to make little mini-me's of most organs. You take a bit of tissue out, you grow it. It'll make chiefly something that looks like an embryonic tissue. So here's the embryonic brain. Basically, you can recapitulate embryogenesis of most tissues in the dish. However, what we really understand about organ biology is this, and recapitulated in the dish, and this, which is what goes wrong in the adult tissue during homeostasis. So we understand disease very well for most tissues, and we understand the first initial steps. But how to grow an organ, we have absolutely no idea. How do you get from this, well, not that, you don't want to produce that, but how do you get from this to this? Now, in this, obviously fairly didactic cartoon here, they've made it seem like you can just simply grow incrementally bits of tissue. Of course, we have no idea what processes drive growth, what stem cells drive growth, what releases stem cells for proliferation, how you do it regionally, and then how do different tissues then coordinate the growth. So I think as a developmental biologist, I always get puzzled when people say, well, we know everything about developmental biology now, and now we're all stem cell biologists because that's what we need to do. In fact, we only know so little about, well, we know a lot about a little bit, which is what most scientists do, basically, but we know a lot about the first stages of development, very little about anything else. So I think that's where the frontier of developmental biology is, personally. So my little bit that I've studied over the last, 20 years now, is the first steps of embryogenesis of the myotome. And I want to use the myotome as a system to study growth. And the embryonic myotome in the fish is very simple. So if you've been watching MKR, where they cook their salmon steaks, you'll see this, the the outside of the salmon is a darker colour and the inside is pink. That's fast and slow separated. They come from different regions. The slow comes from a non-sebatic compartment that migrates to the outside and forms these blue fibres. The rest of the remainder of the embryonic myotome comes from the posterior compartment of the somite and direct differentiation. And the anterior provides the growth during the larval phase. It undergoes a rotation event, forms a single cell layer on the outside, and growth is driven by calling in these cells for differentiation. So here they are, here. In a tomogram, you'll see just in this quadrant here, one of these stem cells coming in, or then a progenitor cell being called in to grow the myotome in here. Here it comes. This is what we've called the external cell layer, a very imaginative name because it's on the outside. And here you see the stem cells marked with this PAX marker, PAX 7. Yes, here we go. And it forms a squamous epithelial layer on the outside of the myotome marked in red and the stem and progenitor cells marked in yellow. So basically that's everything we knew about, rather depressingly, of 20 years of work from my international colleagues and myself and my colleagues in my lab. And I asked, well, we know then how to make this thing, this tiny little thing to scale. How do we make this thing, the adult musculature? And how is it done? Now, in you and I, muscle growth is a very episodic thing. So post-birth, theoretically speaking, no mammal, or most mammals, do not add any more muscle fibres. So you just grow your fibres by the process of something called hypertrophy, which is what Gordon studies across the road mainly. Hyperplasia in fish occurs continually. So you see there is a shift between, here is the size of the fibres in cross-sectional area. Here are the number of fibres. You'll see there, of course, there is a shift where fibres do get larger and the addition of new fibres decreases in rate. But a fish makes muscle throughout the entirety of its life, unlike us. So it's eternal hyperplastic growth. And we asked, well, where do these fibres come from? So we used a fibre maturation assay with fluorophores, which use DS-Red and GFP. Those of you who've used DS-Red know that's an annoyingly slow maturing fluorophore. For us, it takes a couple of days actually to reach, after it's expressed, for it to reach its full intensity. And GFP is a relatively fast maturing fluorophore. So therefore, you can use the combined fluorescence in the red and green channel to work out where an old and new fibre is. So a new fibre would be a green-only fibre, and an old fibre would be a red and green fibre. And you can make heat maps throughout life of the fish and average them. And what you can see here is the oldest fibres in blue are in the middle, and the newest fibres are for the main are on the outside, although you do see some hyperplastic growth. And with this, we're able to show that large diameter fibres are small, so large are old. Large diameter fibres are old, wish I could read. Small diameter fibres are new. So this is just, we can use the size of a fibre as a proxy for its age. So it seemed like we had a zone on the outside of the fish that would correlate with stem and progenitor markers. So here's a stem, another stem marker. It's a PAX gene. There's a PAX gene for everything. So the PAX gene for muscle, a PAX 3 and 7. So PAX 3 in this transgene, you can see marks exactly where the growth occurs. And MIF5, as I mentioned, was a stem and progenitor marker, and myogenin is the differentiated myocytes. It occurs on the outside. So we had an eternal hyperplastic growth zone that existed on the outside of the fish that grew the fish from the outside out. Okay, so I was intrigued how it did that. What was the clonal relationship for growth over time? Is it many stem cells contributing a small number of fibres to growth, or is it single stem cells producing large number of progeny? How do you actually physically deploy your stem compartment? So we used the same system, the Zebrafish muscle bow system, to look at that. And what we actually found was that in the early phases, you'll see, and this is the same fish image at the same level over the course of the entirety of this time. Sorry, not the entirety, it's a taco time lapse. You take the fish out, put it back the next day, image at the same spot. But what you can see is that basically the colours are random, and when new fibres arise next to their neighbours, they're different colours. So this reinforces the fact that in this phase of growth, stem cells contribute randomly to the growth of the myotome. And here again at 5 millimeters, that's also the case. But as you will see over time, and if these segments are the septa, so fish have, as I showed, have these beautiful chevron-shaped septas, which you see when you're eating your fish. They're separated by extracellular matrix to which the muscles attach, and a section catches these. These are all basically compartment boundaries in the true Drosophila sense of the word. And you'll see within an individual compartment, over time, growth becomes to produce a uniform colour within the myotome, such that the entirety of a single segment of a myotome, the growth of it, can be driven by a single stem cell. So this is a classical model of clonal drift, whereas you start off with a large number of stem cells contributing equally, such that, and then one or two cells contribute to growth. In fact, this is perhaps the most dramatic and complete example of clonal drift yet to be described in growth. So clonal drift has been described in homeostatic tissues such as the colon and the gut, and not really thought to be before documented in growth dynamics. So how do you measure this? Well, again, phenomenologically, clonal drift is just what I've showed you. They show you pictures of sections of tissue and they one colour or they're many colours, and that's supposedly enough to convince you that it's occurring. So we had to create a computational mechanism of doing this. So here's what happens in the early phase of a growth of fish. Randomly, the stem cells contribute progenitors. They give random colours. And the way that we measure that, again, is just take the hues and saturations of each individual fibre, we chart it on a colour wheel, and the distance between these should be large, because if they're randomly generated, that will generate a huge interdistance on the colour wheel, and this will be represented by a large bar. However, if clonal drift occurs, such near uniformity of contribution occurs, these cells should cluster in colour by the same type of analysis, and the distance between them, mean distance between them on the colour wheel, should be small. So if you go and do that analysis for every single fibre in a section over many, many, many fish, you can then quantitate the drift, and you'll see exactly at the point that I showed you, you get this shift to clonality. Now, clonal drift has been described, as I said, by Hans Cleaver and colleagues, mathematically to be neutral. That means no clone has any greater chance of generating this drift than any other. It's a mathematically defined parameter. And we wondered whether this was a neutral process itself, so we couldn't do every single colour. Computationally, that would be way too intensive. So we selected two colours, red and green, and asked whether the red and green cells within an individual sample pool acted and fitted the neutral drift hypothesis. And what this graph shows is that within a model, the model that was provided for the colon corrected for growth dynamics, it fits the 95% confidence level of neutral drifting phenomena. So this rather blew my mind, that within our tissues, or even that of a fish, when I say we, I normally mean fish, that we would have a stem cell that would come to make the entirety of your myotome. Just absolutely flabbergasted me. And in fact, I began to wonder whether this is a general phenomena. And if you think about it, if you're really only made from a dozen stem cells in each of your tissues, that changes the way you think about a lot of things, and we'll get to that at the end. But how the hell would such a drift occur? Well, Fong went back to the start and said, well, this external cell layer, which is this eternal hyperplastic stem cell zone, we've got to understand the dynamics of it. So I'm going to make a movie over the course of 3 days of every cell division within that to try and understand where the stem dynamics come from. And he did that, and really, I'm just going to show you this movie, if I can get my cursor back somehow. Come to me. Is it on the screen? There it is. Come back. And what he found was a series of divisions relative to these septa. So I mentioned these horizontal myosepta, which are ECM-rich regions, which intuitively you think would be important because ECM is a very important niche building. So there's one here called the horizontal myosepta, and one here called the vertical myosepta. And what he was actually able to show that the vast majority of proliferation events occurred next to this vertical myosepta, and I'll show you the quantitation of that. And there were two types. They were both asymmetric and symmetric divisions. And then there was a series of divisions that he's showing here within the myotome that generated growth for new cells within the myotome. So he basically charted all these behaviours over several days, And he basically came up with these maps. But the main thing I wanted to reinforce is that the horizontal myoceptor, the cells there divide very, very rarely, whereas the ones at the vertical myoceptor divide continually. And very rarely you would see a septal cell divide and migrate to the vertical myoceptor. So this was a classical low dividing potential stem compartment generating a vertical myoceptor associated transit amplifying compartment. That was what he thought of had in his head. And I said, well, the movies are all very well and good. You can chart all these different divisions and you can put them into a framework, but you have to show me that those cells are low cycling. And the way he chose to do that was to use label retention. So label retention is a property of low cycling stem cells. If you don't divide, when you're given a label, it doesn't dilute over time, whereas the rest of the progeny will. So using heat maps here of label retention of an injected fluorophore, red being high, blue being low, you'll see that the vertical myoceptor indeed is a region of label retention. And so then we went and specifically looked at every single marker we could think of. We wish we had the single cell up and running then, and we could have, but we had no marker to sort these specifically other than the label retention, which is difficult. And we just did candidate gene approach. And we came up with one transcription factor that Fong had actually worked on previously, and it was beautifully and uniquely and specifically restricted to the low cycling stem compartment. And we think this is a stem marker. And so when we took this homeobox gene and deleted it genetically, we saw that the stem cell zones are completely abrogated. We got very different divisions, comparing these colours here and here to here and here, just shows you the divisions are very different. And very importantly, he was able to show a complete lack of label retention in this particular mutant. So we think this mutant affected the stem cell dynamics of this compartment, and we were very intrigued by this, and we wondered how it did that. And one of the things that really was very telling is we took this marker myogenin and put it on embryos and said, well, if myogenin is a marker of differentiation and we're losing the stem compartment, perhaps what's happening in these cells are entering the cell cycle and differentiating precociously, and that's exactly what Fong saw. So the marker here is normally never expressed in external cell layer cells because they need to be regulated in terms of producing rhythmically the formation of growth cells. However, when you look at the ECL with this marker, it's incredibly upregulated in these cells, both at 1.5 days and 3 days. So the deficit in label retention is due to the commitment of the low cycling stem progenitors to differentiation ectopically, and this produces ectopic differentiation within the stem cell layer. Perhaps most amazingly, when Fong took these mutants and asked whether they underwent clonal drift, and here is the phenomenon again in another set of experiments where you drift to clonality. When he looked at these homeobox mutants and said, and this gene is expressed throughout the somites early, and it's beautifully placed and then restricted specifically to the compartment, the stem compartment, you'll see that drift is completely abrogated in these mutants, and you can see this beautifully in the images here. And then when you do the studies to say whether, well, what happens to these, you see it clearly doesn't fit the 95% confidence limit anymore. But if you go back to the images of the previous slide and count how many cells ectopic differentiate and put that back into model, 100% accounts for the drift. So the cells are not drifting anymore because they're lost to differentiation, basically. Okay, so how does that sort of was head-scratching, because here we had a neutral drift phenomenon that no longer happened when you actively mutated a gene. That's kind of a bit of a head-scratcher. How do you have a neutral process driven genetically? So we wanted to know how MEOX1 could regulate the cell cycle specifically, because that's where most of the action is in regulation of stem cell compartments. Everyone drills down onto the cell cycle machinery. So MEOX1 is also called growth arrest homeobox in vascular biology, in adult vascular biology. So when you have a remodeling phenomenon in your vascular, when you have a change in pressure and you need more vascular cells, MEOX1 is downregulated because it actually inhibits vascular cells, specifically at G1. Now, G1 is the stem cell modality of quiescent stem cells. They're locked in G1. That's nearly every tissue-resident stem cell population, including the ones I just showed you, have a G1 arrest. So we thought that's pretty simple. Meox-1 will be doing the same thing in growth-specific stem cells as it is in quiescent stem cells, as it is in the adult vasculature. It'll be locking down G1. So I said, fine, we'll just put this system, this dual Fucci system, which is the system where we can measure the in vivo dynamics of living cells and their cell cycle status based by the degradation properties of different fluorophores. And what he actually came up was not red, but blue for the Meox-1s, which marks the G2. I said, well, this is completely abrogated in the Meox-1 mutant, as you would expect. I said, well, this might just be something wrong in this setting, or these cells might be cycling too fast, you might not be catching them. And I said, what you need to do is assay every single cyclin gene or every part of the cell cycle and tell me which of the things that are upregulated or downregulated to form this arrest, thinking that he would be one arresting, thinking that cyclins, because this was just a poor assay for what we were looking for. And blow me down was that after doing this very extensive study of every single cell cycle regulator, the only genes that were specifically upregulated in Meox-1 mutants were G2 cyclins. And he'd absolutely nailed it, and particularly cyclin CCNB1, which is a known G2 cyclin. CCNA2 does both G1 and G2, but together they're G2 specific cyclins, you could say. And I said, well, look, how does this work? Is it a direct regulation? So he went in and did an attack-seq across the genome and looked specifically at the Meox-1, looked specifically at the CCN1B1 locus, found a peak that was regulated appropriately, that contained Meox-1 binding sites, and found that these regions chipped beautifully with Meox-1. So what this particular set of studies showed was that we'd isolated a dynamic stem cell zone that was regulated spatially and temporally on the outside of the myotome, such that we had a self-renewing stem cell compartment regulated by the expression of Meox-1, and this produced self-renewal by specifically inhibiting CCNB1 and held our cells in G2. And these were low cycling, and they were called into action by yet to be described signals. In the mutant, and this provided us a clonal drift, again by a mechanism we don't understand. So in the Meox-1 mutant, this inhibition of CNCB1 does not occur. You get cell cycle progression, they proliferate but don't self-renew, and you get a failure to undergo clonal drift. So many things in this study really puzzled us, and I think it's a really fascinating area now to go on and try and study these global dynamics of stem cell regulation and growth. As I said, I think this is a huge area. However, there are a few things I thought more globally might be interesting to discuss. So the fact that organs have stem cells that drift to clonality is really intriguing in terms of cancer stem cell hypothesis. No doubt if you've got a dominant clonal growing cell, one assumes that this could well be the template for cancer stem cells. And if you could identify that and actually prove this hypothesis, it would be a fairly major finding. However, the really interesting thing I think about our studies, it came out roughly the same time as this paper by Alex Shear and the Shinendura group, using this Gestalt mechanism. So there's been a number of CRISPR-Cas barcoding lineage analysis is done in a variety of organisms. This was one of the first. But actually what this does was just you tag every single cell in the embryo and then say, how are those cells related clonally to each other in the adult? A very powerful system. And there's now ones that do this as well as looking at single cell sequencing along the lineage. There's just a beautiful paper in Science, if you've got time to read it, about doing that. Amazing paper. And these are all where these lineage tracing techniques are going, not just doing lineage, but also do single cell sequencing of every single lineage state along the way. So by doing this, what these guys found was that amazingly, most organs in a zebrafish are clonally related to themselves. So that suggests that that what we've described as drift within the myotome is actually a general phenomenon that happens in every single organ of your body. And so therefore, this really does emphasize the importance of understanding the dynamics of growth in terms of stem cell populations for a number of disease settings. And the last thing is that I want to emphasize that there are horses for courses. Not all stem cells use the same interaction with a cell cycle to regulate their entry or growth or activation. In this case, we have a G2 arrest of these growth-specific stem cells, whereas adult quiescence, I've mentioned, is associated with G1. And of course, it makes sense that G2, which is not considered such a deep arrest, would be associated with growth, which would be on a hair trigger, responding to cues for growth. But it's intriguing that, that we could potentially regulate growth differentially to regulating activation of the stem cell compartment. So I'm going to stop there, and I hope I provided you with a snapshot of where I think regeneration biology is going, the consequence of stem cell populations in different contexts, and where I think the field is moving. But really this was done, I said, chiefly by 2 graduate students, the first talk by David and Fong, and support from these characters here. And if you ever have any questions about my work, I'm freely available to talk about it. I'm around afterwards for the students or whoever wants to talk to me. But yeah, thank you for the invitation to speak.
Well, thank you very much, Peter. That was fantastic. Now, as chairing this session, I've got a couple of things I need to do. The first is I need to do what I have repeatedly forgotten to do, which is to, of course, give the welcome to country, which is to acknowledge that we are meeting on the lands of the Wurundjeri people of the Kulin Nation, pay my respects to their elders past and present. I'm sorry that I forgot to do that earlier. Now, first of all, students, there is a gathering with Peter afterwards for lunch. Don't forget that. Second of all, I'm going to open the floor now to students. I think we've got a couple of honours students who had to ask questions, who indicated that they would like to ask questions. So if you are one of those students, please, or if you're any other honours student for that matter, or any other student for that matter, please feel free to raise your hand.
Or anybody in general.
Right, down the front. Take us away.
Just on an evolutionary basis, why do you believe that mammals have lost or never developed the extent regenerative capacity?
Yeah, so there's a couple of emerging areas that are intriguing in that. To be honest, I think that certain lineages for trade-offs we don't understand do that. There's a really interesting set of observations that neonatal mice do have a broader regenerative capacity that has a very strict cutoff in temporal phase. So neonatal mice, Enzo Perello, who works over there, over both places, over there and over there, showed that if you do injury in a mouse heart as a neonate, it will regenerate up to a certain point, and a very sharp cutoff that seems to actually correlate directly with the maturation of the immune system. So one hypothesis is that the more elaborate, complex immune system that we have as mammals may somehow prevent the regenerative capacity because it's required to do other things in wound repair. That's a fairly predominating theory at the moment. I actually really do like the lineage-specific loss, just the fact that we lose capacities sometimes for reasons that we can't appreciate. And the more I work with the immune system in the fish, the more complicated it gets. So I'm not so sure. I think the jury is very much out as to why. Yeah.
Earlier you were talking about trying to differentiate skeletal muscle stem cells in a dish, and you said that you haven't been able to do that, and that might be because of the niche. So I was wondering if you looked further into what the niche is that would be required to differentiate those cells in vitro.
Yeah, so that's one of the big thrusts of the lab, and hence are in some of the Rudnicki work that we believe it's the extracellular matrix cues that are coordinated through the dystrophin glycoprotein complex, such as laminin, that's the key player. So we have several laminin mutants that show a reduced number of stem cells and a reduced proliferative capacity, and when we inject just plain laminin from the sarcoma, you can actually increase regenerative capacity. So we think the niche is built on polarized signals that surround laminin biology. Good question.
Sabrina, sorry.
Just going back to the first half of your talk, I was wondering if there are no uninjured cells surrounding the injury site, if any stem cells are still able to migrate to that site.
So if it's just completely wiped out, there aren't any uninjured cells, what happens? They still come because we think actually the macrophages do that as well. They're less, not completely, there's less cells that come when you delete macrophages, but Yeah, we don't exactly know the signal. We actually think it's probably the dead and dying fibres releasing factors that attract the stem cells themselves. But yeah, we have never seen a wound. We make very big wounds. If you make them too big, the fish does struggle to recover. And intriguingly, if you ablate macrophages and you do a needle stick injury, they'll never recover. They'll die. We don't think it's the uninjured fibres drawing in the stem cells. We think it's the dead fibres releasing factors, or their proxy through the macrophages.
Down the front.
I'm not sure if this was mentioned, but do we have an understanding of the asymmetric differentiation mechanism of satellite cells?
In terms of what actually derives the asymmetry, or?
Yeah, like what happens and what does that?
Yeah, well, that's the big question now. There are a lot of factors like NUM that have been implicated in notch regulation and generating asymmetries. FGF signalling is believed to coordinate the amplification, but these are all on fibre analyses. But yeah, the biggest player appears to be notch. The MAP kinase pathway regulates it. Yeah, there's a number of pathways that have been implicated in mammals. Yeah.
Right up the back.
So my question's in terms of embryonic generation and post-embryonic regeneration. So apart from the triggers themselves, such as macrophages, which I assume wouldn't be relevant in embryonic generation, how similar do you think those processes are? So you talked about slow twitch fibers being activated first in post-embryonic regeneration. Do you think that, for example, is the way it also works in generation, and can we learn much from that process?
Yeah, that's a fascinating question, and of course we lose a lot of our competitive advantage when we move into the adult fish because we don't necessarily have the optical clarity, although we are working on that. We have done adult injury models and they respond less slowly, but they respond very similarly. And I think there are two different sections. So embryonic development can regulate itself, so you can cause massive injuries in an embryo and it will grow itself. No one understands that at all because we're all brought up as developmental biologists that there's a signal here and a signal there, and in between is the message. And how the hell do you if you suck out half the head, how do you regrow it in an embryo? That is amazing regenerative or regulative capacities rather than regenerative. In the larva, it's not like that. It's sort of halfway between the embryo and the adult, but you're right to question whether all the same signals will be the same, all the same signals will exist, or whether larval regeneration is a bit more like the embryonic one. It seems to be, for what our context, and we did work very hard on this, to be pretty much identical to the adult scenario.
I'm going to cut it off there because we are at 1:02, so please join with me in thanking Peter for a really stimulating insight.